Hello guys, I'm here making another biochemistry video, this time on the citric acid cycle, and we're going to spend quite a bit of time talking about the pyruvate processing that happens leading in to the citric acid cycle. This stuff about the mitochondria is just important because when we were talking about glycolysis was happening out in the cytoplasm of the cell. As we're moving to the citric acid cycle, we're going to be moving to what we call the matrix of the mitochondria, which is this innermost part within the inner membrane of the mitochondria. I'm just going to skip ahead through a couple of slides to here. I really like this part of the PowerPoint describing pyruvate processing. I think the visuals are really clear, but let's talk about what's going on here. So before we can start the citric acid cycle, we have to take this pyruvate, and remember we have pyruvate coming out of glycolysis, and we need to change it into acetyl-CoA. Because that's what we're going to consider more or less our starting point of the citric acid cycle as we begin to learn it. So pyruvate itself doesn't actually enter the citric acid cycle. We have to make this acetyl-CoA first. And we're going to use this pyruvate dehydrogenase complex to do that. A complex is just a group of enzymes. So that's what this is representing down here. We have 3 different enzymes making up our complex that's going to take this pyruvate and it's going to convert it to this acetyl-CoA, and we're going to talk about how it actually does that. Now the names of these enzymes are right here, but while I talk about it, I'm just going to call them E1, E2, E3 for simplicity's sake. So remember, in our pyruvate dehydrogenase complex, we have the 3 enzymes that we just talked about. We also have 5 coenzymes. Yeah, coenzymes. 3 of which are shown here right now. This thiamine pyrophosphate, which we can call TPP. We have this lipoic acid and the FAD. And remember to go back and review what these vitamins are, okay, I have my TPP, that's actually vitamin B1. And then know if it's an essential nutrient or not. That's going to tell us if we need to consume something with that vitamin in it to allow this enzyme complex to work. So we're going to go into step 1. What we're going to do is we're going to take this pyruvate, here's pyruvate, and we're going to attach it to the TPP of E1. That's all that happens in step 1. We're just attaching the pyruvate to the first enzyme in this complex. Moving on, we're going to decarboxylate. So remember, D is usually taking something off, and we're going to be taking off carbons. And we're going to take them off in the form of CO2. So we're just going to chop this bond off right here. This CO2 is just telling you that what this is. And remember, anytime we produce CO2 throughout any of these metabolic processes, we're just going to breathe that out. When you exhale, you're exhaling mostly carbon dioxide. This is one of those places where it's coming from. And the reason we're going to do that is for the next step to occur. If you're ever confused, well, why did we do that? Why did we take something off? Why did we add something? Why did we change the location of something? It has to usually do with it's important for the next step. Sometimes we don't need to understand the importance for the scope of this class, but for the next step, we need to remove this CO2 so that we can rearrange the molecule. So before we saw a double bond between carbon and TPP, we're going to make that a single bond. We're going to add a hydrogen. That's where that word protonation comes from. We're adding that hydrogen. And the reason we're doing that is so that we can transfer this over to enzyme 2. So this is our lipoic acid, which is one of our coenzymes. And we're taking the acetyl group, that's the name of this part right here, and we're attaching it to that coenzyme. So E1 is done. We're now attached to E2. So what's going to happen next? we need to take this coenzyme A. We're going to use this acetyl group and coenzyme A to make the acetyl-CoA. So what's going to happen is this acetyl group is going to get transferred to coenzyme A. So we're going to see what that looks like right here. Here's that coenzyme A from before in the blue. Remember, coenzyme A is pantothenic acid. I think that's spelled wrong, but that's vitamin B5. And I took that acetyl group, I'll show it to you again, here's that acetyl group, and I stuck them together, and that's how we make acetyl-CoA. So that is what our end product of pyruvate processing is, that we need to go into the citric acid cycle. But we're not done. It would be really inefficient if we stopped here, because notice what we have here. This is where the lipoic acid used to be, but now it's dihydrolipoate. Let's compare this structure to the original structure. So if we stopped here, we couldn't keep using this enzyme over and over and over again. That would be kind of a waste. So the next couple steps are going to kind of be done to recycle our pyruvate dehydrogenase complex. We've made the acetyl-CoA already. We've already processed pyruvate. We just need to reset this system. So what we need to do to reset the system is we're going to take these hydrogens off of dihydrolipoate and we're going to attach them to this FAD. You can see that here. We've returned this to being lipoic acid. Now our only difference between the original complex and what we have here is that this is no longer an FAD, this is an FADH2. So what are we going to do? We're going to transfer one more time. Those hydrogens that were here are going to go here and here to NADH and H+. These are going to go to the electron transport chain. Remember in my last video, I told you anytime you see NADH, NADP, that's a pH, or FADH2, those are all what we consider our electron carriers. They're going to be vital in the electron transport chain that we're going to learn at the end of this week. So now our enzyme complex is reset to its original state, ready to be used again, and we've processed pyruvate into acetyl-CoA. And remember, back in one of those steps, we had carbon dioxide as a byproduct of these reactions. And NADH, of course. This is important to be regulated. Anytime we talk about a metabolic process, we're going to spend the most time talking about deregulation. So let's look at some ways that we can regulate just this one enzyme complex to speed up or slow down the making of energy. So one way is direct inhibition. We can have the molecule NADH or ATP bind directly to the first enzyme in the enzyme complex to inhibit or slow down the process of changing pyruvate into acetyl-CoA. And if we think about this, it kind of makes sense. What are the products of this process? I know we don't actually make ATP in pyruvate processing itself, but ATP is going to be a downhill product from here. We're going to make more of it in the citric acid cycle. We're going to make more of it in the electron transport chain, and the overall process of changing pyruvate into acetyl-CoA is to make ATP. So if we have so much of it available that it has the time to come over here and bother this enzyme, it's probably true that we don't need to be making more. Same idea with the NADH. If I have so much NADH that it's not busy in the electron transport chain, it can bind here and say, we've got plenty, slow down. Now we can talk about some indirect inhibition. So in indirect inhibition, the molecules themselves that we're going to talk about are not binding directly to the enzyme like we saw before. They're going to bind to a separate enzyme. In this example, we have pyruvate dehydrogenase kinase. When these molecules bind to pyruvate dehydrogenase kinase, it's going to take a phosphate group and attach it to this enzyme. This phosphorylation, or adding a phosphate group to enzyme 1, is going to inhibit or slow down the process. Same idea as this first example, we just have an intermediate step. The molecules that can cause inhibition in this way are, once again, ATP and NADH, but we also can include acetyl-CoA this time. Once again, this falls into our logic. If we've made so much acetyl-CoA that it has time to bind here instead of going into the citric acid cycle, that means we have enough energy, we can afford to slow down or inhibit this process. So we're going to bind these molecules to this enzyme, and it's going to stick its phosphate group onto enzyme 1 to slow it down. In an opposite manner, ADP and pyruvate can bind to the same enzyme as before, and that's going to dephosphorylate enzyme 1. Opposite idea of what we were talking about before. These are what I'm going to, for a lack of a better term, call the reactants. ADP means that we need to make more ATP. That's the thing that we build up when we've been using lots of energy. Pyruvate, if we have lots of pyruvate, some of it's got to get shifted into the citric acid cycle. So when these molecules bind to this enzyme, it's going to take that phosphate group that we had up here and pull it off so that this enzyme speeds up. Last example that he has is, once again, a dephosphorylation to activate this complex, but this time we have a new enzyme. We have pyruvate dehydrogenase phosphatase. So the molecules that can bind to this enzyme are calcium and insulin. So if calcium and insulin bind to pyruvate dehydrogenase phosphatase, it's going to tell that enzyme to take this phosphate group off and activate the enzyme and speed up the process of processing pyruvate. So that's really all you need to know about pyruvate processing. What are the steps? What is the enzyme complex that does these steps? What are its coenzymes? What are the names of the enzymes? And how is it regulated? Now that we have made acetyl-CoA, we can move into the citric acid cycle. So we're going to talk about the 8 steps of the citric acid cycle. We, because we're humans, want to think of this as a step 1, step 2, step 3, ... But remember, it's a loop. We can enter into this loop from multiple different places depending on what our starting points are. Because we just finished talking about glycolysis, we often like to think of pyruvate as our starting place, and that's what we're going to the way we're going to reference this moving forward throughout most of my video. But we're at the very end going to talk about other ways and other things that can enter the citric acid cycle. So do remember that this is a loop. When I say step 1, that's kind of just so we can know what we're talking about and where in the cycle we're talking about it. So we're going to pay attention to the intermediates, the enzymes, and any byproducts or important notes to make of each reaction. So when we have acetyl-CoA, remember that was our product of pyruvate processing, the first thing we're going to do is make it into citrate. And I think the enzyme names in the citric acid cycle are really easy to memorize if you know the intermediates. If I'm going to make citrate, I'm going to need a citrate synthase. Remember, synthase is something that makes something. So this enzyme's name means something that makes citrate. Easy enough. Things to note, it requires water. And when we use this acetyl-CoA, we're going to cleave off that coenzyme A that was attached to it before. This coenzyme A can be reused again to process more pyruvate. Moving on, we're going to take that citrase and we're going to make it into isocitrase. So remember, iso is going to refer to an isomer. And if we pay a little bit of attention to the structure of this molecule, we can see this enzyme is just moving an OH group. It's moving it from this carbon to this carbon. And this name is a little less intuitive, but the enzyme that does it is iconitase. So we're just repositioning the OH group. Next step is we're going to make my favorite intermediate, probably one of the most important intermediates, alpha-ketoglutarate. So to make alpha-ketoglutarate, we are going to have to dehydrogenate isocitrate. So it's really fitting that the name of this enzyme is going to be isocitrate dehydrogenase. And whenever you see a dehydrogenase, you know that one of our electron carriers are going to have to be involved. In this step, it's going to be NAD. So we're going to take a hydrogen, this one specifically, off of isocitrate, attach it to NAD to make NADH. Also, in this step, it's important to know that we're going to take off a CO2. This is going to happen spontaneously. We don't actually have an enzyme doing this. It has to do with the shape of the molecule and thermodynamics, but this CO2 is more or less just going to pop off, and whenever we see CO2, we know we're going to breathe that out. Moving on, we're going to take our alpha-ketoglutarate, and we're going to make it into succinyl-CoA. So, to make succinyl-CoA, we need to dehydrogenate again. You're going to see a big theme in the citric acid cycle of dehydrogenation reactions. It takes up most of the reactions in this cycle. So if we're dehydrogenating, we know we need an electron carrier involved. Once again, the culprit is NAD, which is going to be turned into NADH. So we're taking a hydrogen off. So when we do this, we're going to take a coenzyme A. It could be the one from earlier. It could be a different one. Doesn't really matter, but we're going to attach it to the bottom here. And that's important for the next step. Once again, we're going to lose another CO2. This CO2 is going to come off. It's going to make space for this coenzyme A. This step is something important to highlight because it is at this point that any carbons we had from pyruvate are now gone. Let's go back and talk about where we lost those carbons. One carbon from pyruvate we lost during pyruvate processing, all the way back here in step 2 when we decarboxylated pyruvate. So by the time it enters the citric acid cycle, it's only down to 2 carbons in acetyl-CoA. We lost one of those carbons here. We lost another here. So every carbon that was in that original glucose molecule was turned into 2 pyruvates. And when those 2 pyruvates go into the citric acid cycle, any carbons that we started with in glycolysis ultimately get turned into CO2 that we're going to breathe out. So from here on out, it's all about getting as many electron carriers as possible to make the most energy in the electron transport chain. So moving on from succinyl-CoA, we're going to make it into succinate. So for succinyl-CoA, we're going to take that coenzyme A back off. We're going to recycle it. And we're going to have what pretty much the leftover products from that. And this little CO2. That's what we're seeing here. There should be 4 carbons total. 1, 2, 3, 4. In the process of this whole recycling of coenzyme A and making succinyl-CoA into succinate, we're able to transfer an organic phosphate group onto GDP to form GTP. This itself can be used for energy, or we can transfer that into ATP. So it is important to know for every 1 pyruvate, we're going to get 1 ATP, or at least 1 ATP equivalent. You're going to say to yourself, isn't this a metabolic process? Shouldn't we be forming energy? The purpose of the citric acid cycle is less to directly form ATP. Remember, like I've been saying over and over and over again, we're looking for the formation of those enzyme carriers. This is just a little bonus. So now we have succinate. And once again, we're going to have a dehydrogenation reaction. We're going to dehydrogenate succinate to make fumarate. We're going to need an electron carrier to do that. This time, instead of NAD, it's going to be FAD. That's a different vitamin, but it has a similar function. So when we take our hydrogens off, they're going to go on to they're going to go on to FAD, which will make FADH2. That's going to go off to the electron transport chain. Now we have fumarate. Fumarate, the enzyme that's going to convert it to the next intermediate is fumarase. Fumarase. Remember, ASC is a good sign that it's an enzyme. So that's how you know fumarase is the enzyme and fumarate is the intermediate. It's going to take some water and it's just going to throw it on, and just by adding that water, we get a new intermediate called malate. From malate, now at this point, we're going to dehydrogenate one more time. So we're going to need a malate dehydrogenase. And we're going to use NAD as our electron carrier again. And that dehydrogenation is going to make oxyl acetate. Sometimes you're going to see this as OAA because it's really hard to pronounce. So now we just need to take this OAA and recycle it so this can continue being a cycle. So if I look at the big picture, where did it go? We can see here that our citrate synthase is going to take that OAA and that acetyl-CoA from before to make citrate. I do have a mnemonic for both the intermediates and the names of the enzymes that catalyze their reactions. There's going to be a theme where some of my mnemonics are a little bit weird and maybe inappropriate, but this is, it's good because I'm not an actual professor. This is just so you remember it. So for the intermediates, I have can I keep sex slaves for my office. C for citrate, I for isocitrate, K for ketoglutarate, S for succinyl, S for succinate, F for fumarase, M for malate, and O for oxaloacetate. Can I keep sex slaves in my office? Some people add a please at the end to represent pyruvate. I don't like that. I think it's a little misleading because pyruvate, once again, needs to be processed into acetyl-CoA to enter this process. And then for the enzymes, I have an innkeeper, C's, sassy, flirty, men, cooking. I'm not going to go through all the letters. Once again, just like I did with glycolysis, I find it more effective to either memorize the enzymes or memorize the names of the intermediates and use your knowledge of those to fill in the blanks about what the enzymes are or what the intermediates are, instead of trying to memorize both mnemonics. I think if you use a little bit of logic and memorize one or the other, you can use your knowledge to come to an answer of what that missing piece of information is. All right. The last thing we need to talk about, remember I said this is a cycle. We sometimes say step 1, step 2, ... But we can actually enter the citric acid cycle from many different points, especially using amino acids. We're going to talk about this much later in the semester, I assume, but this is a good time to address what amino acids can go into what part of the citric acid cycle. This is one of those things where it's good to have it memorized so you can just recall it. So when we actually get to start talking about these processes and how they're converted into something to join the citric acid cycle, you already know what amino acids are going where. So the first category we have are amino acids that can be converted to pyruvate. So just like any pyruvate formed from glycolysis, if we make an amino acid into pyruvate, that's going to get processed and enter the citric acid cycle in the form of acetyl-CoA. So those enzymes, or not enzymes, those amino acids are going to be alanine, glycine, serine, threonine, cysteine, and tryptophan. I have a mnemonic for this one, and it's all girls see those crazy types. So AL, AL, GG, SE, SE, TH, TH, CC, and TY, TY. All girls see those crazy types. Alanine, glycine, serine, threonine, cysteine, and tryptophan. Our next group is going to be amino acids that can be converted into alpha-ketoglutarate. Remember, we saw alpha-ketoglutarate. I said that one was my favorite down here. So we can convert amino acids into the substrate, and then they can be converted to 6-acyl-CoA, so on and so forth, creating more electron carriers and energy. So these amino acids are glutamate, glutamine, histidine, arginine, and proline. I have another mnemonic for this one. It's go, get, high, after, passing. G, 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 H, I, H, I, A, and P. Do whatever you have to do to memorize it. But like I said, it is one of those things where as much as I hate memorizing, I even had to have this memorized in other biochem classes because it's important to be able to recall it quickly. Amino acids converted to 6-acyl-CoA. We have valine, isoleucine, methionine. For these shorter lists where it's 2 or 3, I don't have mnemonics. At that point, I don't think anyone really needs a mnemonic. It's only 3 you have to remember. If you want to make a mnemonic, go at it. So 6-acyl-CoA we saw right here, so we can convert these amino acids into this molecule and it will go through its loop. Oxaloacetate, remember I said that was our OAA. We have aspartate and arginine. He did have a note here that this is the enzyme that converts asparagine. I don't really know what this enzyme is. I'm sure we'll talk about it more when we start talking about how to do these conversions. Amino acids can be converted to fumarate. We have another 3, phenylalanine, tyrosine, and aspartate. And not last but not least, we have acetyl-CoA, we have leucine, tryptophan, isoleucine. We will talk more in the future, I'm sure, about what it means to be ketogenic. I wouldn't worry about what this word means right now, but do know that leucine can be converted into acetyl-CoA. And he also has these notes here. We saw tryptophan earlier here, and we saw isoleucine here. So it is important to note that one amino acid could be converted into more than one intermediate of the citric acid cycle. It's going to depend on what materials are available in the cell, maybe what cell we're in, which intermediate it will become. For acetoacetate, we have leucine, lysine, phenylalanine, tyrosine, and tryptophan. This is not for the citric acid cycle. This is involved in cholesterol synthesis, but it's the same concept. Just it's important to have this for recall reasons. I do have one more mnemonic for you. This one is Lucy lays petting tiny tigers. You can use these mnemonics if they help you, or you can flashcard memorize them. That's the end of his PowerPoint on the citric acid cycle. Definitely focus mostly in on pyruvate processing. But I think it will be helpful for you to have all 8 steps memorized of the citric acid cycle. Like I said, I don't think it's as hard to memorize as glycolysis. My one advice for you, if you are trying to memorize these metabolic pathways, and this is not my own idea, I got this idea from a tutor, and it has been super effective for me, is take the time, especially if you have a whiteboard, this can be fun. Draw out the metabolic pathway. This could be the citric acid cycle. This could be glycolysis. This could be pentose phosphate pathway. Draw out the names of the intermediates and the enzymes and any important coenzymes or byproducts that we've highlighted. Don't worry about the chemical structure. That's not important to this class. It's just the names that are important to us. Take the time, draw it all out. Go back, erase a couple things at a time, See if you can fill in the blanks. Then go back, do it again, erase different things. My roommate and I have been going back and forth doing it. It's really effective. It's a great way to memorize things super quickly. And if you can kind of do that, even if you don't have scratch paper on exam, I promise you, it will be much easier for your brain to be able to recall what happens in this step, what enzyme is used in that step. Enjoy the rest of your snow day, and have a great day.